So, going on now, he says the conclusion of a common ancestor for humans and mice is virtually inescapable. See that term again, kind of like Ken Miller used. You know, there's no doubt about it. But once again, our friend, the logical fallacy. Okay? He's affirming the consequent. He's saying if humans and mice share a common ancestor, then we should observe these shared AREs. Well, shared AREs are observed between humans and mice. Therefore, humans and mice share a common ancestor. Again, just because the grass is wet doesn't mean it rained. And just because we share similar AREs doesn't mean we have a common ancestor. We have a common designer God who probably put them there for a very good reason. And we need to figure that out. We need to do that. And as creationists, you know what? We have a reason to do that because we believe that God designed it that way. But the evolutionists, they don't really have a reason to do it because after all, it's just an evolutionary leftover, right? Why bother? That's prohibiting science. Whereas we as creationists are progressing science because we see that as the result of God and we want to study it and we want to figure out what it's doing. Now, the very last argument he says here, he says this is an overwhelming challenge to those who hold to the idea that all species were created ex nihilo. I don't know of any creationist that believes that all species were created ex nihilo. Okay? This is very commonly heard. This is called a straw man argument. Okay? So he's going to basically set up an argument that we don't even use and then destroy it. But we don't even use that argument. So that's the problem with it. And this scientist here says, hi, I'm a creationist. He's, you know, for the little puppet here, hi, I'm a creationist. I don't believe in science. Everybody knows science is real. What else don't you believe in? Well, creationists don't believe that animals change. But science shows us that animals do change. Absolutely. I totally agree that animals do change. I never said God created all species ex nihilo because his word doesn't say that. It says he created kinds ex nihilo. He created the different kinds, which we believe is about the family level of classification, not genus, not species, but family. Okay? So again, that's the problem with that type of argument. He's setting up a straw man. So what can we say? Well, we can say that junk DNA is not junk, and we really need to rename that. We've got to figure out a better term to use for that because it's just very problematic. Shared AREs are not evidence for common ancestry. That is a logical fallacy, and those that argue who God wouldn't have done it that way are being completely arbitrary. And a common designer equals common design. That's exactly what you would expect to see, and that's exactly what you do see. All right, now we go to question, I'm losing track here, 4. Does gene duplication add new genes with new functions to the genome? Again, this is one we're seeing a lot of in an increasing amount.
Welcome back to Truth Seekers. And you know, that is what you'd expect to see. I mean, God, who's omniscient and omnipotent, would use the best system for everything. And so you would expect to find similarity. He's not going to use the best for the humans and a lesser system for some other. I mean, he would do the best kangaroo he could make, and he would do the best gorilla he could make, and the best elephant, and the best human. And
of course, since living creatures share the same need for nutrition, then we have to have a chemical system that will process food in basically the same way, so that if I eat fruit or some other animal eats the fruit, it will provide nutrition to the physical sustenance of the animal. So there have to be similarities. Well, God could have done it a different way, but it's really very practical. So you really expect these kinds of similarities. It's what's expected from a very efficient God. So really, it's not outside the expectation of Scripture. Is precisely what we would expect from the Almighty, Holy Creator God, who's created everything when He said it was very good at the completion of creation.
We were talking about, during the film a little bit, about the functionality of these repetitive elements and the fact that now, that study she quoted was in 2007.
Yeah, I saw 2007 there. I was surprised to see that, and I would never want to get into a snowball fight with Dr. Collins. He has a very high reputation. I can't agree with his presuppositions, but I've read, and maybe it was since 2007, but it was within the last 2 years, that perhaps 93.5% of the human genome is now recognized as being functional. And what a lot of that, okay, let's go back up. The way this whole junk DNA came out was about 30 years ago. There was a Dr. Ono, and it was shared by Crick and a few other people. Certainly our good friend in England,
Dawkins.
The God Hater, yes. But that you would expect then, of course, 90% of the DNA would be not functional because it would be remnant. It would be left over from all the deep, dark past of evolution, and it's just kind of hanging around. It's not useful now, and eventually the continued evolutionary process will drop it off. Well, and the reason they came up with that, with the 10%, and Georgia Purdom said 3%. That's kind of surprised me because I'd always heard that about 10% was or is functional in doing what? In synthesizing or in synthesizing protein. And 30 years ago, when they were looking at this stuff, they only understood, or the only purpose that they saw of the DNA was to synthesize protein. But in fact, now all of this recent research is showing that many segments of the DNA are copied into RNA that are not those. Usually, the RNA that's been understood in the past was to synthesize protein, but there are many segments of the RNA that are used for what's called micromanagers, to carry out many processes going on, not in the nucleus, but in the cytoplasm, in the part of the cell outside of the nucleus and inside the cell wall. Many processes. And the comment that I saw by a British scientist about a year and a half ago, they said, well, we're up to 93.5% and we're still finding things out. So maybe we're going to find out that really all of this DNA is functional. Now, that's just one point. And that didn't surprise me, and it did not surprise Bible-reading Christians, because the God of the Bible that we know is not a God who makes junk. And so even when people like Ono and Dawkins and Crick were calling this junk, we just knew, as Jesus said, time will tell, kind of, or the truth will tell out in time. That's one thing.
And go ahead.
Well, I want to mention another thing, and you may have seen that there were many repetitive segments. Well, I don't.
By the way, we have about 2 minutes left, I think.
Okay. Like well, I'll just do it very quickly. If you want to Google it, you'll find it in Wikipedia about fractal structures. F-R-A-C-T-A-L. It's a certain kind of structure that, if you have a certain structure, you have another structure inside that has the same shape identically. Inside that, another and another and another. If you go into Wikipedia, you'll find examples of fractal structures. And some recent research published maybe within the last 6 or 8 months indicates that, in fact, the DNA is stored in a fractal structure form. And it's very interesting. Well, what might that imply? Well, I've had work with relational databases. And many times, if you have a database, if you want to access information efficiently, you don't store that information only one time. It's like maybe having one set of car keys in your pocket and one set of car keys in the trunk. You may need them one place one time, not the car keys, your total set of keys, and another place another time. So in efficient databases, many elements are repeated identically in order to provide effective rapid retrieval. And so in essence, that's what DNA is. It's a very efficient data system. So I'm sorry, just that was quick. Maybe still too long.
That's fine. By the way, we're going to be on the next few weeks we're going to be on